Good day, I'm Dr. T and welcome to my office. So today I would like to talk about biomacromolecules that don't fit neatly into one category because they fit into two categories. And I'm going to be focusing on those that would fit at least in part into the carbohydrate category because those are kind of the ones that we see the most, or at least the most interesting from my point of view. So when we're naming these, you'll have two names for them, and the root or the last name in the line is the one that's the bulk. So if I have a proteoglycan, then the proteo is short for protein, glycan short for carbohydrate or sugar, then it's mostly sugar but some protein. If it's a glycoprotein, it's mostly protein with some carbohydrate stuck on it. So let's look at first the proteoglycans. Proteoglycans are going to be, at their core, carbohydrate. And these are going to be examples of things like chondroitin. However, this carbohydrate is going to be very, very heavily adapted. It's going to be a polysaccharide, but some of the alcohols are going to be replaced with sulfate groups or amino acids, and definitely some peptides being put on here. So it's mostly a carbohydrate. It's mostly a polysaccharide, but it's heavily modified with amino acid and protein constituents attached to it. And it can be a fairly complicated structure. Chondroitin is used heavily in extracellular matrices. It's incredibly hydrophilic, so it's going to be able to attract lots of water, create this very hydrated environment that's going to provide some nice lubrication. And to be clear, extracellular matrix is the space between cells. So you're not just cells. You've got spots that are between cells, and it's going to have things like chondroitin or collagen or the actual minerals in your bone. That's all extracellular matrix. Now, it doesn't have to be between carbohydrates and proteins. You can also pair up with lipids. Lipopolysaccharides are found in gram-negative bacteria. Now, otherwise they would be fairly dull, except for the fact that, one, gram-negative bacteria can be a source of infection, and lipopolysaccharides are recognized by our immune system and treated with extreme prejudice, to the point where if there's enough of them present, you can enter into toxic shock. Essentially, toxic shock is your immune system responding so aggressively that there's enough collateral damage that you can't survive. So the response is basically, with all force, at all costs, kill these bacteria, and all cost ends up being your life, potentially. So septic shock, not something you want, typically present in massive infections. So cases where, say, a soldier goes into war, gets shot, gets wounded, imagine like World War I, and then massive infection sets in because they're not able to get to medical attention in a prompt fashion, because once again, you've seen the movies on World War I, absolutely horrendous. But you can also see other cases. There was a case in the 70s where some young women were leaving their tampons in for incredibly long periods of time, and that led to bacterial growth and toxic shock. And there's another really interesting case that's kind of in the background. Injectable pharmaceuticals, so, shots and whatnot. Those have to be checked to make sure there are no lipopolysaccharides present in them. Because you can kill bacteria, but when you kill the bacteria, it's not like they magically disappear. They get ground up, and the parts of the bacteria, after it's exploded or shredded or in other ways degraded, are still present. So you can have an injectable that you've sterilized, so, IV solution, shots, whatnot, but it still has lipopolysaccharides present, and your body will respond to those, especially if they enter the bloodstream, which is like, whoa, how'd you get here? And your immune system doesn't recognize the fact that there's no bacteria. It's just dead parts of bacteria. And so it will respond with extreme aggression, and once again leading to toxic shock. The way we address this is predominantly by making sure that it's not there, and we screen our injectables with horseshoe crab blood extract. Horseshoe crabs, ancient species, really weird things. They literally have blue blood. One of the proteins in their blood will react with lipopolysaccharides, forming kind of a gel. So you extract that protein from horseshoe crab blood, and you can use that as an analysis for lipopolysaccharides. Okay. So the last two examples have been polysaccharides. Let's look at oligosaccharides. So oligosaccharides are going to be shorter. They're the oligomeric form, the ones I haven't really talked about so much. And these are quite often used for identification purposes. Think shipping labels and flags and whatnot. So you'll see easily things like glycoproteins. These are going to be proteins. They're going to have oligosaccharides on the surfaces of them. And in fact, for humans, lots and lots and lots of our proteins are actually glycoproteins. This is one of the major jobs of the Golgi apparatus. And so this is going to then help the cell know, okay, this protein really is mine. I don't need to go nuts about it. This protein then, needs to get shuffled off to this particular location. They, in many respects, act like a packing label. Oh, you need to go to the Golgi, or you need to go to the rough endoplasmic reticulum. Ship it over there. That kind of scenario. Additionally, you can have things like mucins, which are mostly protein, but they've got lots of carbohydrates on them. They're modified, and if you're thinking mucins sound like mucus, yeah, that's because they are. These are then excreted into orifices and whatnot to provide this nice sticky goo that can be flushed out and, go from your lungs, take the dirt, kick that out, and either down the stomach or, out one of the assorted openings in you, and thus cleaning various body parts and keeping the lungs dirt-free, which, of course, you don't want dirt in your lungs. So you got a couple options. You can use them to make the proteins much more hydrophilic, which is a common approach in the mucins, or you can use them for labels. Additionally, you don't have to attach them to a protein. You can go for, say, a lipid. So glycolipids are going to be an oligosaccharide stuck onto what's typically a phospholipid of some flavor, and that way you can stick it in the cell membrane, and this is going to once again add labeling. Things like, hey, I'm a cell, you can grow on me. Which is a popular approach. Other ones of simply identifying what the cell is, or communicating that, hey, I'm part of me, kind of scenario. This is what the ABO blood groups are. So for our cells, specifically blood cells, but by no means just blood cells, we have glycolipids all over the cells. These glycolipids will have, an oligosaccharide on the surface, and your immune system learns not to attack it. Now, you could actually have 3 different oligosaccharides. You could have the O oligosaccharide, which is actually a truncated version of the other 2. So no one's immune system should be recognizing the type O antigen, or that oligosaccharide that could be attacked by the immune system. Now, some folks will have a little bit of an extension on theirs, and that would be the A antigen. So it's going to be the O part plus the A part. And antibodies in the bone marrow will learn not to attack those. And so if you have the A antigen, you do not have antibodies against it. And thus, if I wanted to put more blood in you because you were leaking for some reason, then I could put type O blood in you because you don't have antigens against the core part of that oligosaccharide, or I could put type A blood in you. But if I put type B blood in you, then you would get antibodies against the type B blood because your cells have not learned not to attack it because you don't have that natively. Alternatively, you could have the B antigen and not the A, in which case it's simply a reverse. You could get B blood, you can get O blood, but if I put A blood in you, it'll attack it. Of course, other individuals have both the A antigen and the B antigen. They're going to be on different glycolipids, but you still have both. Your immune system learns not to attack either, so yay! A blood, B blood, A, B blood, which is both, O blood. You can take it all, and not a big deal. By the way, when it does come to blood type, the plus and minus is a different antigen. I believe that's a protein-based one. And there's more going on, but that's usually good enough to figure out what will kill you and what won't kill you if we squirt it into you. Because, once again, good old septic shock and general rejection. So with that said, that's a brief rundown of oligosaccharides as well as all the weird combinations you can do with carbohydrates and other things. Actually, I shouldn't say it's a rundown of all of them. There's more than one can shake a stick at, but those are kind of the major ones. So I'll talk to you later and have a wonderful day.